Our final speaker is Professor David Julius, Chair of Physiology at the University of California, San Francisco, educated at MIT and trained at Columbia. He has devoted his career to unraveling how we sense the world by discovering the receptors that detect temperature, pain, and irritants like chili peppers. He revealed the molecular basis of somatosensation. For his work, he was awarded the 2021 Nobel Prize in Physiology or Medicine. His research continues to inspire new approaches to understanding and treating chronic pain. Please join me in welcoming Professor Julius, who will present gut feelings, probing mechanisms of visceral pain.
Oh. No, thank you very much. Elaine, I have a question for you. If I start smoking cigarettes to ease my stress, will my hair grow back? All right. It's great to be here. You know, I started my life in the lab anyway, as a nucleic acid biochemist. So being here to honor the center and Severo Ochoa is a great honor. And thank you, Paulo, for inviting me. We were postdocs together at Columbia, different labs, but on the same floor. We probably haven't seen each other for 35 years, so it's a real pleasure to come here. I've enjoyed this meeting, and I liked our discussion yesterday, the roundtable, about teaching our kids how to be curious and use scientific facts. And it reminded me of this conversation I had with my son a few months ago. We were in a sort of a crowded touristic part of San Francisco, and all these people are taking pictures of one another and of themselves. And I said, you know, it's interesting that you don't see people with selfie sticks anymore. And he said, well, you know, Dad, you're a biologist. You should understand this. Remember when we were in Stockholm, we went to a talk by this guy, Svante? And I said, yes, about human evolution. And he said, well, you know, probably over the last 20 years since the cell phone's been invented and everyone's taking pictures, the human arm has evolved to be longer. So and the selfie stick has become vestigial. So thank you for teaching my son about human evolution. And I knew he was trolling me, but I looked at him and I said, I'm very proud of you, my son. So anyway, biology touches everybody. Okay, so today I'm going to tell you about some work that we've been doing in the lab to understand mechanisms of visceral pain. So the focus in my lab really is to understand sensory systems at large, but to understand pain in particular. And I'm sort of the representative for this consortium that we call the gut group, which consists of Holly Ingram's lab. Holly's here today. Jim Bayer, who's a pediatric GI person at UCSF, and Stu Brierley's lab, who's a fantastic visceral scientist, pain scientist at the University of Adelaide, and of course, people from my own lab. So I want to start off with this just to remind us that, you know, as with the term cancer, we use the term pain as a unimodal thing, but like cancer, pain is, you know, a collection of different disorders and diseases and syndromes. You know, migraine pain is certainly mechanistically distinct from post-herpetic neuralgia, from osteoarthritic knee pain, et cetera. And I think one of the important goals now that we understand some of the players involved in somatosensation and pain sensation is to use these tools to understand how different chronic pain syndromes differ, because until we do, we can't come up with, I put here pharmacological treatments, but any kind of other treatments that may involve devices, et cetera, to really understand how different pain syndromes may respond to distinct or in some cases to similar treatments. So I think one of the goals is to sort of pick your favorite system and try and get to those questions. And the systems that you choose, of course, depend upon their approachability experimentally, and also some of the intrinsic questions, you know, at the level of cell and molecular biology that, you know, we happen to find particularly interesting. And over the last few years, this has sort of led us to focus our attention on visceral pain, namely gut pain in our case. And why have we focused on that? Because it affects a very large percentage of the population. About 10 to 15% of people report being affected by some sort of chronic visceral pain. These are things like, you know, irritable bowel syndrome is probably one of the more common ones. And like many chronic pain syndromes, in this case, it's say post-inflammatory or post-infectious. One of the most common instigators for IBS is something like a serious bout of food poisoning. And then what happens is, you know, all the sequelae of that, the inflammation, tissue damage will resolve, but people can be left for for days, weeks, or years, or forever having this sort of chronic debilitating syndrome, even though there's no longer evidence of tissue damage. And this is an example of many what we call functional pain syndromes, where people complain about having chronic pain and debilitating pain, but there's no longer any sort of physical sign of an injury that might have instigated this. And that leads to a lot of problems in terms of diagnosis and treatment. As I'll talk about today, a main topic is that there's gender disparity in visceral pain. Women suffer from chronic visceral pain much more frequently than men. In the textbooks, it'll say about threefold, but if you talk to GI specialists, like one that said, Holly and I talked to at a meeting recently, she'll say, I have no men in my clinic, it's all women. And you know that this is the case because if you see commercials for drugs or other treatments for IBS, of which there are not many good ones, it always features a woman, and it's often a younger woman in her reproductive years. So something that is relevant to what I'll say. And related to these other points, like many pain syndromes, there's a lack of biomarkers or overt pathology. And the sensory mechanisms have remained obscure, but I think we're starting to close in on some of that. We meaning the field in general, not just us. The other interesting thing about this is that, relevant to many of the things we've heard, is that there's a lot of biology involved in visceral pain. So if you're interested in different aspects of biology, especially you students, it's a place where it's kind of a playground for understanding interactions between immune cells, the microbiome, their effects on the peripheral nervous system, and how these effects lead to sensitization both in the periphery and in the central nervous system. So there's a lot of biology here and a lot of unknowns. And today, I'll ignore a lot of these things for the point of simplicity and probably, to our own disadvantages initially, but eventually you have to sort of fill in all these components to really understand what's happening physiologically and pathophysiologically. All right, so here's the group and a number of people from my lab up on top, from Jim's lab, from Stu Brier's lab, who runs the visceral pain unit at the University of Adelaide, and Joel Castro, one of his senior people. And the work I'm going to talk about today is really spearheaded by Archana Venkataraman, a great fellow in Holly's lab, and by Eric Figueroa, a wonderful postdoctoral fellow in my lab, and how they've sort of collaborated to understand mechanisms that are associated with sex differences. And Holly's lab's really interested, as many of you know from talking to her while she's been here, in understanding mechanisms of sex differences and how this is relevant to women's health. And after 30 years of marriage, we decided it was safe to collaborate on something. So our interests have come together over this topic. All right. So we're interested in the gut. And like you've heard, so some people, Elaine might tell you that the skin is the largest barrier surface in the body. It's not, it's the gut. But it won't grow hair back anyway. But in any case, we're interested, the gut, of course, is this hollow tube. We're interested in the interface between the lumen, where all the food and bacteria stuff live, and the rest of the intestinal layers, which includes the mucosa, which I'll talk about, and then all the interstitial nervous and muscular systems. One thing I want to point out about this that's really sort of captivated us that I won't talk about too much now, but other people in my lab are really interested in this structure. The gut is such an elaborate and a beautiful structure where you have the stem cells living in the crypt, much like, hair follicles, and then they migrate up into the villus, and you have this continuous turnover. And topologically, it's a really complicated and beautiful structure. And there's so much signaling that's going on here. And as people who are interested in signaling mechanisms, we're interested in understanding how signaling occurs in this very elaborate architecture, and how different cell types talk to one another. Not something I'll talk about too much today, but we've been started trying to develop techniques, live cell imaging techniques, to really understand the temporal and spatial dynamics of signaling within the gut epithelium. And what we focused on, if you look at the schematic where you have, so of course the luminal space where all the food goes through and the bacteria live, and if you eat sushi, you might experience a worm or two, is separated from the neural network and the muscular layer by this, epithelial sheet that carries out transepithelial transport of water, nutrients, and ions, et cetera. There's a really complex nervous system in here. Some say as complex as the CNS, and it consists of intrinsic motor and sensory neurons. We're mostly interested in the so-called extrinsic primary afferent neurons. These are the ones from the brainstem of vagal origin, or the ones from the spinal cord, the spinal primary afferent neurons that send projections from these central regions into the intestine and go way up, as I'll show you, into the villus, and interact with cells within the epithelium and other cells, intrinsic motor and sensory neurons within this mucosal region. So we're interested in these primarily because those are the ones, particularly the spinal ones, that carry nociceptive information or pain signals from the gut to the central nervous system. So we want to know how these neurons interact with the epithelial surface and how they interact with all kinds of irritants that impinge on this surface, both from the outside, namely the luminal surface, and from the inside in the mucosal region. Now, if you and how they send signals, of course, to elicit behavioral responses such as visceral pain, nausea and food aversion, and anxiety, which is really a combination of activating cells within the spinal and the vagal afferent system. All right, so if you look at this epithelial layer blown up, what you'll see is that most of the cells are sort of these, what I would call nondescript, transport cells, the epithelial cells that carry out water and nutrient transport. But among those are scattered these rare cells that are of neuroendocrine origin that are actually electrically excitable. They express voltage-gated sodium, calcium, and potassium channels. And so they're sensory. What's kind of interesting about them too is that, I gotta go back, is that they're rare. They represent less than 1% of all the intestinal epithelial cells. They're diverse. There are 8 or so subtypes depending upon how you parse them molecularly. And as I said, they're excitable. They fire action potentials, and some have what we think are anatomical attributes of a presynaptic cell, mostly because they have the capacity to release neurotransmitters and neuropeptides. Among these, I'm going to tell you about 2 types. So we've been interested for several years in this one cell called the EC cell, or the enterochromaffin cell, is a subtype of these excitable enteroendocrine cells. And people have been interested in these for many years because they are serotonergic, and they actually produce and release the majority of serotonin in your body. They account for about 90% of the serotonin in your body, and almost all the serotonin that's peripheral, except for a few neurons in the gut that may, intrinsic neurons that may also produce 5-HT. But this is where most of the peripheral 5-HT comes from. And people like Gershon and others have shown over the years that the serotonin that's released from these cells activates primary afferent neurons, such as these spinal neurons, that are marked by genes such as capsaicin receptor TRPV1, and most notably by the one member of the serotonin receptor family that's an excitatory ligand-gated ion channel called the 5-HT3 receptor. And it's this pathway that some have believed is important for carrying nociceptive information from the gut to the CNS. And here you can see these cells in so here's a piece of the epithelium rolled up on a Q-tip. They call this a Swiss roll. When I talked about this in Sweden, I call it a Swedish roll, but in any case, it's really called a Swiss roll. And you can see the little green dots, like stars at night, are the very rare EC cells. And EC cells are among the most abundant of the enterochromaffins, the enteroendocrine cell types, but they're still rare. Here's a blow up of it. You can see how beautiful they are, and they're sort of decorate both the villus and the crypt. And they're what I think you want to take away from this slide is that they're small but mighty. These cells elicit very powerful physiologic effects, but they represent a very small percentage of cells in the gut. All right, I'm going to tell you about another cell type today, which you may have heard more about, called the L cell. You may have heard about this because it's a peptidergic cell that releases peptides involved in metabolism and digestion, like PYY or GLP-1, the body's own Ozempic, CCK, et cetera. And the role of these cells have really been studied mostly in relationship to metabolism, satiety, et cetera. And I'm going to tell you about how these cells interact with one another in the context of visceral pain and sex differences in visceral pain. So let me tell you a little bit about what we've learned about EC cells over the last, 5 or 10 years. And that is that they're excitable. And when Nick Bologna was a fellow in my lab and worked with Jim Baer when he was a fellow in Holly's lab, they characterized these cells in detail biophysically and pharmacologically. And what they found is that indeed they express, voltage-gated channels, so they're excitable, as I said, and they express receptors for a number of relevant agonists that are relevant to the detection of noxious stimuli in the gut. So these include, for example, what we call orphan olfactory GPCRs like OLF558 and OLF78 that you'll hear about a little later. These are receptors shown by many other groups to detect short-chain fatty acids, things like acetate and isovalerate, that are produced by bacterial metabolism of things you eat, like carbohydrates, simple carbohydrates. In people who have certain types of dysbiosis like IBS, some of these things like acetate or isovalerate are either elevated or are products that can exacerbate the symptoms of visceral hypersensitivity and pain. The other one is this channel that Mike told you about a little earlier that he used in flies, but that in mammals called TRPA1 is what we call the wasabi receptor. It's not really temperature sensitive in mammals, but it's exquisitely sensitive to irritant chemicals, namely any irritant that's has electrophilic properties. So things like wasabi and other legumes that you eat have compounds like allyl isothiocyanate and all these isothiocyanates. Onions have a lysine and all these reactive chemicals, electrophiles, that activate this ion channel that depolarizes the cell by allowing sodium and calcium to enter into the cell. And so a number of ingested irritants, and in fact, some inflammatory mediators like some prostaglandin derivatives also activate these channels and lead to cellular depolarization. And once activated, this, of course, leads to the release of serotonin from the basolateral side of the cell to activate the 5-HT3 expressing primary afferent neurons that go to the spinal cord or to the brainstem to elicit nociceptive responses. And one of the things that we're also interested in is that there is sympathetic input to these cells. You can see that beautifully histologically, which I won't show you. And our lab and Art Bader's lab at Mayo has shown that these cells also express adrenergic receptors on the basolateral surface. And so one of the questions is this sort of a feedback mechanism whereby stress regulates visceral sensitivity? We all know that when we're stressed, we have increased, visceral sensitivity. And people who suffer from IBS, anxiety is one of the sort of comorbidities of this situation. So I don't have anything to say about that today, but that's something that we're very interested in. So here's one of those beautiful cells, again, in an organoid, in this case, an intestinal organoid, where you can see it labeled by TD Tomato. And here you can see it just firing spontaneously because it's loaded with a calcium sensor. And so, whoops, I don't know what happened to this. This disappeared. This is what happens when you can't use your own computer. Anyway, this what in this little box was an illustration of what I'm just telling you about of an EC cell. And the questions that we want to ask here are, do EC cells regulate visceral hypersensitivity? Can we really show that that's the case? Are they an important part of a nociceptive circuit in the gut? And most importantly for this talk, does this EC cell mucosal afferent circuit that I showed you on a previous slide play a role not only in visceral sensitivity, but if it does, can it account for any aspect of sex differences that are observed in humans and potentially in mice? All right. So let me start by addressing the first question, and it gives me an opportunity to tell you about the assays that we use in this system. So one of the assays is sort of equivalent to what people have classically used in the pain field in cutaneous pain, where they use something called the skin nerve preparation. Here it's a gut nerve preparation, and Stu's lab is really one of the masters at this. And you take a piece of the mucosa and you just lay it out in this recording chamber, and you extrude the sensory nerve fiber through a this gap, so you could record from it. When in this case you apply, say, pressure to the mucosal surface to activate the sensory nerve fibers, which are mechanically sensitive, and you can do a lot of pharmacology this way. And if you do that, in this case, what we did was, again, things are not showing up here, but in this case, what we did was we used a mouse in which we expressed the DREAD receptor in the enterochromaffin cells that are expressed within the mucosa. So we can specifically activate the EC cells by putting on a DREAD agonist. And when you do that, what you see is that you heighten the sensitivity of the primary afferent to pressure, showing that the EC cell is talking to the primary afferent neuron and it's enhancing sensitivity in this preparation. So, and the other assay that you can use to interrogate the system is more of a whole animal behavioral assay, where what you do is you basically give a mouse a colonoscopy. You put a balloon into the colon, and as you inflate the balloon and distend the colon, you initiate a protective reflex like you would if you had a colonoscopy, where you hunch and the abdominal wall muscles constrict, and you can measure that with a pressure transducer. And in this assay, you measure what's basically called a visceral motor response to colorectal distension, and in fact, that's the way that IBS is diagnosed in people. And when you do that in this animal, you can see that if you activate the EC cell specifically with a DREAD agonist, you very robustly heighten the behavioral response to colonic distension by activating this EC cell mucosal afferent circuit. So this shows that this is really a very active circuit in how the gut epithelium talks to the primary afferent neuron to exacerbate sensitivity of the visceral system. Now, one thing you'll notice in the slide is that all these experiments are done with male mice. And Holly once asked Stu, well, Stu, have you ever looked at females? And he says, no, everybody always looks at males because they're afraid of variable estrus effects, et cetera. And when Stu did that in the same assay, what he found is that, in fact, females have a higher gain, essentially higher response than males in this ex vivo mucosal afferent nerve fiber response. And you can see that quantified here. And if you do this with an animal in which you remove the ovaries to take estrogen out of the system, what you see is that the OVX animal now behaves more like a male. So you've diminished sensitivity to in this mucosal afferent circuit to, in this case, a mechanical stimulus. And you can see that quantitation here. And if you do that in vivo using this visceral motor response, you see the same kind of thing, where if you take the ovaries out of a female, you make it more male-like by diminishing its sensitivity to colorectal distension, as quantified here. And what's most amazing about this, if you take the animal in which the ovaries have been removed and you give it estrogen just for 6 hours, you can restore the sensitivity of the gut to colorectal distension. And again, here's the quantification of that. And the other result from this that's quite striking is that tonic EC cell afferent activity is higher in females. That's the lesson from this slide, and it's driven presumably by estrogen. And if you take a male mouse and give it estrogen, you can make it more female by heightening its sensitivity to colorectal distension, so it looks more like a female than it does a male. And again, all these experiments were carried out by Archana, who's really been sort of spearheading this aspect of the project. Okay, so we know that estrogen is somehow changing the gain setting for females in terms of colonic sensitivity, no, not working. Okay, and I think it missed a couple of slides here. No? What's going? Oh yeah, all right, let's see, sorry. I'm trying to go back, but ordinarily I'd feel pretty stupid, but given the history of this little remote. All right, let's try this again. All right. Yeah, I'm trying, but that's not responding either. All right. Okay, so I think this has skipped a few slides, but the real question is where is estrogen working in this circuit? Okay, so if you look at estrogen receptors, what you find is that ER alpha, estrogen receptor 1, is expressed in the colon. And interestingly, it's primarily expressed or predominantly expressed in the proximal distal colon compared to, say, the ileum, which is most relevant to what we're talking about because that's where most of the nociceptive information is going to come from. And you can see this here in this quantitation from Holly's lab. All right, so estrogen acts to ER alpha to enhance visceral sensitivity, and you can see that here because if you take an intact female and you knock out the estrogen receptor specifically in the epithelium using a villain promoter, you can see that it looks more male-like. It has lower sensitivity to colorectal extension. And if you take an OVX animal and now try this trick to see if you can restore sensitivity with by injecting estrogen, it no longer has an effect. So all of these effects are now driven through ER alpha to enhance visceral sensitivity. And the question is, where is ESR alpha expressed? What kind of cell types is it expressed on in this colonic epithelium? And we assumed, based on all of that we had done, that this receptor is going to be expressed in EC cells, because that's where all the action is, and that's the part of the nociceptive circuit that we think is really driving pain. And so we spent a long time sort of staring at these things and realizing that if you look at ESR alpha and you look at it, see if it's co-expressed with TPH1, which is a marker of the serotonergic EC cells in the gut, you see no overlap. And this sort of threw us for a loop for a while. But if you then try other markers, what you find is that the estrogen receptors instead expressed very beautifully in the subset of cells that express the peptide PYY, which, as you may remember from one of my earlier slides, are the so-called L cells, not the EC cells, but another group of cells that are actually more rare, maybe present at only 10% the level of EC cells, but are quite distinct nonetheless. And here you can see again the quantitation of the overlap, where there's 100% overlap with the cells that express PYY or CCK, some degree with GLP-1, and not at all with TPH1. So there's no overlap with the EC cells. There's plenty of overlap with the peptidergic cells that are categorized as being EC cells. And again, you see this most prominently in the distal colon. So ER alpha is expressed by L cells, and to our surprise, not EC cells. And what Archana found is that when she gave animals estrogen and looked at that 6-hour time point where you get this very robust physiological change in the setting of the visceral sensitivity, what you see in the blood is that you get a release of PYY into the blood. Interestingly, you don't see much of a release of other peptides, most notably GLP-1. And people like Tui Schwartz have shown that GLP-1 could also have actions on other cells, like EC cells within the area. So, but this suggests that the real, perhaps, conduit between the L cells and the EC cells, if there is one, is through the release of peptides like PY. All right, so estrogen increases circulating PYY, but not GLP-1, which also leads just to an interesting cell biological question, which is if you have a peptidergic cell, how do you have an agonist, for example, that releases one peptide and not another? And what are the sort of cellular molecular mechanisms for that kind of control? And that's not something that we have an answer to yet. All right, so let me say a little bit about PYY and what we know about that. So PYY is found in two forms in the gut and other places. It's found as a peptide. The peptide that people have looked at mostly is a processed peptide called 3-236. That's the one that people have studied mostly in the context of metabolism and satiety. But before it's processed, it's made as a precursor 1-36. And these have actions at different receptors. So there's two PYY receptor subtypes, NPY1R, for which PYY 1-36 is an agonist, and the 2YR subtype, in which either 1-36 or 3-36 is an agonist. And it turns out that this is the 1R is the one that's predominantly expressed in the gut. And if you ask where it's expressed, you can see it's expressed in a few cell types within the gut, within a small number of cells within the gut, and they have almost a complete overlap with TPH1, which says that they are EC cells because they're serotonergic. And you see some expression in some of the primary afferents that innervate the mucosa, but the majority of expression is within the serotonergic EC cells within the gut epithelium. So this suggests that PYY is in fact the functional conduit between the estrogen modulated L cells and the EC cells that we think then are conveying the visceral sensitivity responses to the central nervous system. All right, so to check this out at a cellular level, Eric set up some very nice cellular systems to look at signaling And what he showed, in fact, is if you take an organoid in which you can, in which the EC cells are labeled with GCAM, so you can look at calcium in them, and you put PYY 1 through 36 on them, in fact, you see that they show very robust responses to bath-applied PYY 36. And you can see that here compared to depolarization with potassium. And this activity is blocked by this antagonist called BIBO, which is a selective PYY1 receptor antagonist. So EC cells have PYY1 receptors, and they are functionally responsive to PYY. And the question is, does that do anything to them in terms of their ability to release serotonin? And he asked that by taking an EC cell. Again, you can see its response to PYY or any other agonist by calcium response to GMP. And then you can put what we call a 5-HT sniffer cell next to it that has one of these reporters from Yulong Li's group. So you can look at the release of 5-HT from the EC cell while you're looking at excitation of the EC cell and record that activity here in the sniffer. And you can see that here more easily in the quantitation, where you see that activation of the EC cell, shown here in blue, is temporally locked to the release of serotonin from the cell. And then you just depolarize it with potassium or with 5-HT to maximize the sensors. So when PYY acts on the cell, it definitely releases a bolus of 5-HT and couples to that pathway. All right, so what does this mean physiologically? What it means is that you can circumvent the whole estrogen pathway by just supplying PYY in this, for example, this gut nerve preparation. And if you give an OVX animal where there's no estrogen around PYY, you left shift the dose response curve much in the same way as if you gave estrogen. And again, if you take an OVX female, a strip from an OVX female in this assay and give it PYY. So this is where we use light to activate the system because we have a channel rhodopsin in the primary afferent neuron. So you can shift the sensitivity of the system, whether you activate it with light or with a mechanical stimulus, now by overriding the estrogen responses just by giving PYY directly, saying that PYY is acting downstream of the hormone response. And similarly, in the behavioral assay, if you take an animal which is OVX, and then you knock out the estrogen receptor, so there's no ability of estrogen to activate the system, and there's no estrogen on board, you can again make these animals look more female-like by just providing them with PYY intracolonically. So PYY restores both neural and behavioral sensitivity of the estrogen pathway to female mice. Okay, so and you know, one thing I think, again, one slide didn't come up, and there was a gap of slides in this for some reason, and one of the introductory slides I was going to have is, you know, and you might ask why females have a heightened sensitivity to in this system. And you know, Holly and other people would tell you that's because, especially during reproductive time, and you want to protect the fetus, you have to be very sensitive to what your alimentary canal takes in, and whether you are swallowing things that are potentially harmful, or you have dysbiosis. And the idea is that it's really the cycling levels of estrogen that are most important for mediating this neuroprotective and nociceptive response. And then the idea is that females have a heightened level, but that it takes something else to really trip them over the edge to generate a chronic visceral pain, and that can be some kind of a bacterial infection or some other environmental stressor. So I just want to say that for the slides that are coming up because they were lost in the meeting. Okay, so the model here is that we have these two cells that are talking in a paracrine fashion, both very rare cells. So the question, one interesting question in the future is, how do these cells talk to one another in this very complex architectural environment? But nonetheless, we know something about the pharmacology of the signaling now. Estrogen works on these cells to modulate gene transcription to somehow cause the enhanced release of PYY into the mucosal region to act on the neighboring, or perhaps not so closely neighboring, EC cells to activate NPY1R receptors on these cells to potentiate the release of serotonin, so that you can potentiate the action of this mucosal afferent nociceptive circuit to elicit enhanced visceral sensitivity in this circuit. All right, now, and pharmacologically, we believe that this is true because BIBO, which antagonizes the NP1R receptor, or allosetron, which is an antagonist of the 5-HT3 receptor, in either case, will counteract these effects. So pharmacologically, everything makes sense in terms of this proposed pathway. Now, the other thing that Ochoa and Eric did was to profile these cells, for L cells, in the absence and presence of estrogen, to ask what transcriptional changes might be occurring. And a number of things changed, but one of the things that I want to talk about today that came up is this olfactory, orphan olfactory receptor, one of the ones that I mentioned before, called OLFR78, which we know from the work of others is a receptor for short-chain fatty acids, such as acetate. And you can see that OLFR78 is expressed again. It overlaps with PYY as a marker of L cells. And so Eric asked the question, does acetate activate these cells in this case, in organoids made from the colon or colloids? And what you can see is that in the absence of estrogen, there's not a lot of activity, but when then you challenge these colloids with estrogen, you now develop a much higher sensitivity to acetate. Again, consistent with the idea that OLF78 is being activated as a downstream of transcriptional regulation by estrogen, and it's enhancing the sensitivity of L cells to bacterial metabolites such as acetate. And here you can see the quantitation of that. So the model now is further amplified by saying you have this amplification process and this paracrine signaling process in response to estrogen. And the other thing that now gets put on the surface is increased levels of OLFR78, which makes these cells more sensitive to products of the microbiota and potentially can explain why there's increased sensitivity in the presence of reproductive hormone to the microbiological environment. And this gets back to the comments that I had a minute ago in the slide that was missing about what's happening potentially in women or females when they're challenged with changes in the environment and things that they might eat that leads to a change in their interaction with the microbiota in the combined presence of having high levels of cycling estrogen. And I wanted to point out this paper from this Japanese group where, you know, you do all this fancy molecular biology and make all these mice to look at this. And several years ago, they didn't look at the role of estrogen, but they predicted the interaction between PYY expressing L cells and EC cells just by using some beautiful and simple pharmacology in ex vivo and in vivo systems. So I want to tip my hat to them. All right, so one of the things that I wanted to mention is that, as I said, highly fermentable carbon foods that are poorly absorbed, that have short-chain carbohydrates, are metabolized by bacteria to produce these short-chain fatty acids like acetate and isovalerate. And it's been known for several years, or suggested that that people who have IBS should pay attention to some dietary restrictions that limit the eating foods that have these poorly absorbed short-chain carbohydrates. And so the people have been advised to conform to what they call these low FODMAP diets that limit the intake of these kinds of molecules. And in fact, as Holly always likes to show this slide, there's a very popular TikTok site called Hot Girls with IBS. There are about 10 million subscribers to this, of women who live with IBS, and they have partnered with companies like Belly Welly to, see if offering these kinds of foods, these low FODMAP foods, to people with IBS will actually help improve their lifestyle. And, the question is, what's been the underlying mechanism for this? And I think some of these findings may help explain why certain diets predispose people to visceral hypersensitivity, and how certain types of food or avoidance of certain types of foods can be important in that regard. All right, so in summary, so here's a beautiful picture of, you can see the primary afferents coming deep into the villus and the rare EC cells that interact with them. So EC cells are key integrators of noxious stimuli in the gut, and this EC cell mucosal afferent circuit plays a really important role in signaling visceral pain, both acute and persistent. And one thing that we've shown that I didn't talk about is if you persistently activate this pathway, in males or females, or in males where we've done it, in a dread mouse where you can inject dread agonists and drive this pathway, you will generate a persistent visceral hypersensitivity syndrome that's akin to IBS. So pushing this pathway continuously and robustly is a way to really change visceral hypersensitivity, not just acutely, but persistently. Females show heightened activity of this circuit, which is driven by estrogen, and we presume cycling estrogen is important there. And this paracrine interaction between L and EC cells, we believe underlies a large part of estrogen-dependent sensitization. And this increased acetate sensitivity and PYY release, may drive this enhanced L cell, EC cell mucosal afferent circuitry, again, providing a cogent explanation for how your diet and microbiome feed into this sort of sex differences in visceral pain. And two things that this brings up is that PYY has really been looked at, before this as mostly for its role in satiety and metabolism. But this now suggests that PYY is really, plays a key role as a nociceptive peptide transmitter as well as suggesting how acetate and other short-chain fatty acids and products of the microbiota may, or limiting those kind of things may provide benefit through, say, low dietary FODMAP diets, et cetera. All right, so I think I've mentioned all the people who are involved in this work. I just mentioned the funding agencies. As we talked about, there's a lot of issues going on in the US today. I'm always grateful to the NIH for funding our work. I think work from funding from private foundations is really important and essential, but there's nothing that takes over the role of our federal governments in supporting science, because that's really the contract between our communities and our scientists. And there's a lot happening in the US right now, but as you all know, we as scientists, if nothing else, we're persistent. So we will make it through the next 3 years or 3+, whatever it is. And we will come out the other side of this, and importantly, maintain our wonderful interactions with the global scientific community. So thank you again, and I'm happy to take any questions.
David, thank you very much. Sorry for the voice that I don't have, but very simple question. Is there any difference in proportion between L cells and EC cells between male and female?
Oh, yeah, that's a good question. So is there a difference in prevalence of EC cells or L cells between males and females? And at least when Archana and Eric and Fernanda counted, we don't see any histological difference. So I think it's all in the activity difference. And that was the first thing we looked at. Do females have more EC cells before we even knew about the PYY? And they don't. Yeah, so it's not anatomic, it's biochemical.
So maybe I'll continue on along these lines. With regards to the EC cells, how long lasting are they? Are they derived from the intestinal epithelial stem cells? And it's typically thought that all these differentiated cells are shed,
right?
But that may not be the case.
No, that is the case. So the EC cells, like all the other cells, are derived from the stem cell precursors, and they move up. There's a few interesting things. They move up the villus, and as they move up the villus, they are actually not, they change their properties a bit. So they stop expressing TRPA1 channels down in the crypt, and they start expressing other TRP channels that are sensors for different types of irritants up in the villus. So there's some really interesting dynamics. They do get eliminated, I think slower than some of the other cell types, but their half-life is still, on the order of a month, 2 months. And they turn over. And that raises interesting questions because, we had looked anatomically at some point and suggested that there might be synaptic interactions between primary afferents and EC cells. Koki Tuhara from my lab, who's built these systems to look at the sort of biophysics of transmitter spread, thinks most of the communication, as Gershon and others thought many years ago, is more action at a distance and volume transmission. But there may still be some interactions that we, don't know about. And the question is, if there are interactions, how are those maintained as the EC cells in the epithelium is turning over? So that's an interesting question. Yeah, you.
So it was really interesting to see the difference between the male and the female.
Yeah.
But just getting back to the male in the first place, when you were looking, they also showed this a response which was not as big as the
Yeah.
female. So the nature of this response, how is it actually controlled? So it's not depending on the L cells, or how
Oh,
does it
you
work?
mean what controls the
Yes,
baseline
yeah,
response in
yeah,
males
yeah.
if you increase the pressure and the
Exactly.
I think just the probably just the intrinsic level of serotonin release and their interaction with the primary afferent. I think it's just, you know, the physical nature of the signaling system. And, you know, the more, so you can stimulate the system in multiple ways. The way that it's usually done is through pressure. So there are, you know, piezo channels in some of the EC cells, not all of them, but the primary afferents themselves are pressure sensitive. So you sort of stimulate the system just either by inflating the colon or by pressing on the colonic epithelium. What the EC cell really is doing is changing the gain of that system. So if you take the EC cell out, you'll still see a response. But with the EC, but once you activate the EC cell with isovalerate or some other agonist or, you know, wasabi, then you will change that. So I think a lot of the intrinsic sensitivity is just set by, in this case, the mechanosensitivity of the system, both in terms of the mucosal layer and the afferents themselves. Yeah.
Thank you very much. Very nice talk. I just wonder whether the side effects of serotonin-specific antidepressants can have in women as compared to men has been correlated to this or not?
Yeah, well, so that's a good question. You know, since this is serotonergic focused system, have serotonergic has serotonin pharmacology been used? So allosterone, which many of you know is an antiemetic, it's used for a common treatment for, you know, emesis, but there are also a lot of 5-HT receptors in the gut itself. It's used to treat certain forms of IBS. So there's IBS with constipation, IBS with diarrhea, and it works in women with, I think it's IBS with constipation, only one of those. And the same thing's true for SSRIs. And so none of those things are very effective as you think they might be, but they are used. And whether there's a difference, and there is a difference, like allosterone is not very effective for men who have IBS. So there are some differences. But the real question here also is, will antagonists, for example, the PYY systems or other systems that we know now interact, have some synergistic effect with those things? And that's not yet known.
Just a quick, two couple of questions. So one is, if you superimpose an infection,
Yeah,
H. pylori,
right.
what happens? So
Yeah.
that's my first question. And then maybe for us women here in the room, maybe this is for Holly, that transition right through into menopausal,
Yeah,
postmenopausal,
right.
I mean, some.
Yeah,
some-
actually, yeah, I'll let her answer that. But in terms of bacterial infection, we haven't done that yet to look and see what happens if you change the microbiota. One thing we want to do is just a simple experiment. What happens if you change diet and get, you know, change this FODMAP diet to a high fermentable diet? Okay. You want to answer that question about?
Okay, so that's a great question. Most IBS does affect young women of reproductive age, and their symptoms really do increase along the menses, you know, along the cycle. For perimenopausal women, symptoms also rise. And one of the reasons that we think that happens is because of this massive fluctuation of estrogen. So that's a great question. And I don't think we really have it answered, but that's the best answer I can give.
Quick question, if I may. If it's known whether GPCR desensitization mechanisms may play a role in modulating this overall visceral pain?
Yeah, that's a great question. So, you know, in these transcriptional changes, which we're going through now, we're looking for things that might, you know, GERCs or what have you, that might regulate the G protein pathway. And, you know, the one thing that's weird about the gut is that when Nick and Jim did this initial work on looking at EC cells, and also people like Art Bader, you know, the sort of the canonical signaling pathways, like a GI coupled receptor talks only to GI, et cetera. You know, that seems to be much more flexible here. and the downstream channels are somewhat variable, like different trip channels. So understanding the signaling pathway is really important. It's hard work in this system because the cells are so rare. And even in the organoids, it's kind of a pain in the, well, I won't use the term, but anyway, it's a pain in the colon. But it's, you know, it's hard work to do, but understanding the biochemistry of that pathway is really important. And, you know, something that, for example, people in my lab like Eric are really interested in. Yeah.
So very beautiful, very inspiring. Do you see any change during the development? I mean, and I'm thinking during aging, but also during lactancy, for example. And so the process between.
Yeah, that's a good. Yeah, we haven't looked at that yet, and we should. I mean, it's complicated also in terms of then you have these cells turning over, you know, as Lane said. So, and the other question, you know, what happens in different phases of things like pregnancy, et cetera. And we have not yet looked at that, but those are great areas. So that's something, for example, that Archana is really interested in pursuing. Yeah.
Thank you very much for your talk. I was wondering, because you, just shifting a little bit the focus on the EC cells, you are measuring basically the response of the neurons, right?
Mm-hmm.
In both the mouse and in your in vitro system.
Correct.
And those are also from female and male.
Mm-hmm.
Do you think they can also be responding differently?
Yeah, so that's a great question. That's a great question. It also comes into the other question, related question of what happens after you establish a chronic visceral hypersensitivity. Does that become instantiated in the sensory neuron or later on in the spinal cord or other parts of the CNS? And that was one of the reasons that we set up this model where you can have a dread receptor in the EC cell and stimulate it chronically to generate what we call sort of a colitis-free model of visceral hypersensitivity. So you're not confounded by all the, so the way this is usually done is you put an irritant in the colon, you get all this inflammation, it resolves, and then you're left with visceral hypersensitivity in the mouse. And if you can bypass all that stuff and just activate that circuit specifically, and so that's why we sort of have tried to develop that kind of a model. And then we can ask questions like this, you know, what changes along the way? In terms of the difference between males and females, at least initially, I think that there's not much of a difference in the primary afferent itself, and that most of it's being driven at the epithelium, but we probably should look at that more carefully.